people are more or less back in, or I think. Okay, let's get started again. So next we have Olga Vitek, who's a professor at Northeastern University and expert in statistics, biostatistics, and is also a longtime veteran of these courses. And she's going to explain to us what we need to do in terms of experimental design and statistical analysis. Olga, thank you very much.
Good morning. Can you hear me okay? Is it working? Yes. Okay, well, I'm very glad to see you all here. So my name is Olga Vitek. So I will be talking about introduction to experimental design and statistical analysis. So unlike Alexei's talk, this is not a research talk. So this is a very basic introduction to statistical methods, and some of you may already be quite familiar with that. However, together with the organizers, I feel very strongly that it is important to spend an hour and a half and go over the basic concepts for everything which involves quantitative analysis. And so we will go over that today. And then on Friday, it will be more of a research-focused lecture where we'll talk about how we apply these methods more specifically to the analysis of quantitative mass spectrometry experiments. So for the moment, it will be very general, but I will try to make examples relevant to mass spectrometry as much as possible. So, well, first of all, why do we need to do this? So why do we feel strongly that statistical methods are an important part of the conversation here? And the reason is that variation and uncertainty are unavoidable in the type of experiments that we discuss here. So, of course, there is technical variation, so which really everything which happens before the samples get into the mass spectrometer. So sample handling, storage, processing, all of these things have implications or affect the abundances of proteins which we will quantify with our experiments. Instrumental variation, so elution time, ion suppression, peak overlaps, things like that, of course, also affect our ability to quantify peptides and proteins. Signal processing, and obviously there are many tools already, and you have heard this week, and you will hear more, that different strategies of interpreting the data, they also have impact on the quantitative information. And so all of this is really important, and often we tend to focus on these aspects. And I hear every once in a while people saying, you know, we have so much progress with instrumentation and with data analysis, so if only we can perfect our workflows, then everything will be clear, and then we do not need statistics anymore. And I would like to object to this strongly, because even though we can potentially have really, really accurate measurement systems and really good workflows, what we're actually doing here, we're dealing with living organisms. And so there is always biological variation present in our samples. So there's always a natural variation of protein abundance from one person to another, from one time point to another. And even though we have a perfect measurement system, we still have to deal with the fact that our object of study, the biological systems, are inherently variable. And so our question of interest then is, if I apply treatment to my organism, so is there a change in protein abundance more systematic than as would be expected with this biological variation? So in other words, no matter what we do as far as sample preparation, data acquisition, and data analysis, we always have to worry about the biological variation. And so this is what statisticians do. And so statisticians contribute to this area, well, with the goal of ensuring effective and reproducible research, and we contribute in three ways, at least in three ways. And the first is the experimental design. How can we design the experiments in a way to make sure that our conclusions are relevant to the question that we ask, as opposed to measuring some artifacts which are not really relevant to our question, and how we can do it in the most efficient way. And so this will be a big portion of our discussion this morning. The second contribution of statisticians is data analysis. So now that we acquire the data, how can we interpret the data in a way to really distinguish random variation from systematic changes? And the last part is statistical software. So it's not enough to have the methods and kind of concepts which explain how we should proceed, but we also need to make sure that if I analyze the data set today, tomorrow you can give it to your colleague or somebody can repeat and reproduce the data. So this can be reviewed, critically evaluated, and reproduced. And so while there are many definitions of statistics, you can think of this as a body of methods for making wise decisions in presence of uncertainty, right? And so that's really what we will talk about here. So I will focus on quantitative studies, and so what are the issues which are really important from the statistical perspective. And so this is just to emphasize that this is not new. So the fact that there is biological variation and we need to deal with that. Every time there is new technology, people think that we do not need statistics anymore, right? And so this is just an example from, this was 2011 when RNA-seq came along. And so I don't know if you remember from genomics, so suddenly everybody was analyzing one sample. And so again, if we can quantify more transcripts, so suddenly it comes at the expense of having fewer replicates. And to the point that these statisticians felt compelled to write a manuscript in Nature Biotechnology, which is titled "Sequence Technology Does Not Eliminate Biological Variation," which is silly, but this is a point that needed to be made, that every time we have a better measurement technology, it does not eliminate biological variation. And it's the same thing here. So what I'm seeing a lot in the literature is, okay, we had DDA, then we had SRM, now we have DIA, so we can quantify more proteins, so suddenly again the number of replicates becomes very small. But even if we can quantify more proteins, we did not eliminate the biological variation, right? And so that's what we will need to talk about. Okay, so the first thing I would do is, we have a really big program for this hour and a half, but before we start talking about experimental design and so on, the important thing is, how can we translate the biological or the scientific question that we have into the statistical language? And I think it is very important because when you talk to your computational statistical collaborators, there's always a difference in language and difference in terminology and so on. And it's important to have the right language because then you can communicate your needs more clearly to your collaborators and it will define the overall experimental design and data analysis workflow. So let me start with that. So from the statistical perspective, we can really broadly distinguish methods into three groups, or three goals. So the first statistical goal aims at class discovery. So this would be examples. For example, here you are all familiar with that. This is a heat map. So here the rows would be, for example, proteins. The columns are samples. Color intensity corresponds to the abundance of the protein in this sample, right? And so class discovery would say, okay, show me patterns. For example, show me proteins which have similar quantitative profiles across samples, or show me samples which have similar profiles across proteins. And so essentially you're looking for patterns, you're looking for similarity. And so this is an unsupervised method in that you don't really tell the method what is an important pattern and what is not an important pattern. So you tell the method, go and find something, and it will find something. But if it finds something, it doesn't mean that this something is actually useful for us, right, for our investigation. So another example is principal component analysis, where the axes here are some linear combinations of protein abundances. The dots are the individual samples. And so if the samples cluster together, we'll say, well, they have some similarity of their protein abundances. So the reason why I mention this is that oftentimes, this type of methods are misused. So, for example, if you are doing biomarker discovery workflow and you want to know which proteins are regulated under these conditions or involved in the process of the disease, so then people would look for some kind of similar patterns or maybe use loadings associated with individual proteins and principal components, and they will claim that they found biomarkers. And I would like to emphasize that this is wrong. Because here you just say we found patterns, but some of these patterns can be similarities of random variation. They're not necessarily useful patterns. And so proteins with high loadings or proteins which have similar profiles or somehow change abundances here, they may or may not be relevant to the process of the disease. And this is because the method is unsupervised. So this is useful to visualize the data. This is useful to understand how much information content we have. It is very useful for quality control, but this is not enough to do things such as biomarker discovery. So the second statistical goal is class comparison. So now this is a supervised method in that we have samples which are labeled as healthy, for example, and samples as labeled as disease. And I will just use biomarker discovery. It's easy to talk about this, but you can imagine the same thing if you have wild type versus some gene knockout, right? Or different cell lines or whatever else is relevant. So class comparison would mean that we have two groups of samples, let's say healthy and disease, And here the dots are the protein abundances on the log scale. These are the healthy replicates, these are the disease replicates. So now our question is, on average, across healthy subjects, is there a systematic change in protein abundance as compared to the average of the disease? And so here, the methods that you would use would be t-test, analysis of variance, and so on. And the readouts that you would get would be p-values or posterior probabilities, and so on. And so this is quite useful already because it's a supervised method. If there's a change in abundance on average between disease groups, then you know that the protein was up or down regulated, so it's somehow implicated in the condition. And again, a comment that I often see in the literature is, okay, I have a protein with a small p-value, therefore I found a biomarker of disease. And again, I would like to emphasize that this is wrong. So these methods are useful to understand what is regulated overall, but this is not useful if you want to do biomarker discovery. And the reason for this is that we need to think about the definition of what we mean by a biomarker. So a biomarker is supposed to be actionable, right? So when a doctor sees a person who comes to the consultation, so the doctor needs to be able to look at the readout and decide if this person belongs to the healthy group or has the disease. But what we do here, it's actually something else. We're not looking at the individual patient. We're looking at the averages over all the subjects in the healthy group and averages over all the subjects in the disease group. So this is not the same question, because the doctor does not see the average patient. The doctor sees this particular patient. And so let me just kind of illustrate this a little bit more. So if we have two groups, so here very clearly you have little variability around the mean of each group. So if a patient comes with this value of protein abundance, it's very clear that this patient comes from the disease group. So here the means have a systematic change, and you can also see that from this intensity that the person comes from the disease group. But how about here? So if we have enough replicates in our experiments, so we can be really, really confident in this mean. We can be really, really confident in this mean. So we can claim that this change is not an artifact of random variation, right? It is systematic. It will give a small p-value. However, if we have a new person coming in with this value of protein abundance, this person can be either in the healthy group or in the disease group. So this is not actionable for the clinician. So testing protein abundances for change, right? So testing for differential abundance is not quite answering this question. And so here is the opposite situation. So we have two proteins. So here we can clearly see low value of protein 1 will be, let's say, healthy group. High values of protein 1 is the disease group. The same for protein 2. So here they're both differentially abundant and predictive of the status of the disease. But here's a situation where we have protein 1. We see that the blue dots, so that let's say these are healthy patients, they span the full range of possible values of protein 1. And the same thing is for the red dots. The red dots also span the full range of protein 1. So if you test this protein for differential abundance, you will not have a small p-value. It will not be determined as differentially abundant. The same thing for protein 2. However, as a multivariate panel of two proteins, they're perfectly predictive. So in other words, if we test proteins for differential abundance, they may or may not be predictive. If they are predictive, they may or may not be differentially abundant. So the reason why I'm saying this is that you see already by now that we can direct ourselves to very different types of analysis depending on what question we're asking. Right? And so if I go back here, so the comparing averages is very useful. For example, when we do some exploratory screening studies, we have relatively small number of replicates. We just want to know what is changing overall. However, and so if you're familiar with the fit-for-purpose approach to biomarker discovery, so lower level kind of initial studies could benefit from that. But if we are already in the later stages where we really want to claim that the proteins are useful for diagnosis or useful for some other clinical purposes, we need to do something else. And so this is the statistical goal 3, which is also a supervised goal, which now aims specifically at predicting the status of the individual subject. And so now it would look like that. So if we have two proteins, so let's say blue dots are healthy, red dots are disease, we essentially need to find a decision rule which determines which subject comes from which group. And so one easy thing to do, let's say we ignore protein 1, we only have a cutoff for protein 2, high values are healthy, low values are disease. So it's simple, right? But we can see that some of the disease subjects are misclassified and some are healthy as well. So what we can do, we can have a more complex decision boundary where now we use both proteins and we have fewer misclassified subjects. Or we can have some very, very, very complicated decision boundary which does not misclassify anybody. But this is likely too simplistic. This is likely overfitting our data. It's too describing our selected subjects too well, right? And so the goal is to find a boundary which is complex enough but not too complex. And so the methods that you would use would be logistic regression, I don't know, random forest, support vector machine, deep learning, whatever you want to do, right? So essentially, again, different types of analysis would be used for this type of questions. So the very first point I want to make is when you decide on your whole strategy of experimental design and data analysis, what you need to do first is to ask yourself, what type of workflow I'm doing? Am I discovering new patterns? Am I comparing the protein abundances between known conditions on average? Or am I actually in the business of predicting the status of individual samples? And so all of these three goals would imply very different statistical analysis strategy. So that's a very important point. And I often see people kind of coming to me saying, yeah, why wouldn't we do a PCA and then we will do a t-test and then we will do the deep learning, And so it's just like a mixture of completely unrelated things. And so the very first thing we need to say, okay, let's clarify exactly what type of objective we have scientifically and what type of statistical analysis matches this scientific objective. So this is number one. So for the rest of actually today and on Friday, what I will do, I will focus on this goal 2, where we compare protein abundances on average. And the reason is primarily because this is the objective that I see most frequently in mass spectrometry-based investigations, where we want to do more than just discover patterns without any annotation, right, or any supervision, but at the same time, usually we're not quite ready yet to do like the last stages of clinical investigation and prediction. So let me do this. So what now? But first of all, do you have any questions? It's okay? Okay, please stop me at any time. Okay. So let me now focus a little bit on the experimental design for the purpose of comparing protein abundances between the groups. So if a collaborator comes to me saying, I want to design a study which has multiple 2 or more conditions, and I want to know which proteins are regulated in these conditions, so this is what is actually happening in my mind, right? So this diagram here kind of shows how I would think about this problem. And again, just to keep it simple, let's say we have 2 groups, healthy and disease, but you can imagine any other group that you have in your research, right? So what I'm imagining is that there are 2 populations out there. One is all possible subjects which exist and which are healthy. And the second population is a collection of subjects which have exactly the same characteristics as the healthy subjects with respect to everything that can affect protein abundance, such as lifestyle, gender, age, other diseases, anything you want, right? Except for the fact that these subjects have the disease and these subjects are healthy. So the status of the disease is the only thing which distinguishes these 2 populations. And for all practical purposes, these populations are infinite. Well, they're not infinite, but we can assume that they're large enough that we cannot really deal with them. We cannot enumerate them. So if we want to study proteins in this population, so the first thing we need to do, we need to include subjects into our study. And remember, we were just saying earlier that there is a natural variation of protein abundances from one person to another. So this is our first layer of variation, because if we want to repeat our study later, we will include some other people in the study, and we will not have exactly the same protein abundances in our samples. So this is the first layer of variation. This is the variation between randomly selected individuals. This is biological variation. But this is not the whole story, because next, we actually don't observe this. What we do observe is the readouts from the mass spectrometer, and for the moment, and I think for another while, the measurement technology itself is subject to uncertainty and variation. So this is our second layer of uncertainty, because if we take the same biological specimen from the freezer, we reacquire them on the mass spectrometer, we will not see exactly the same peaks. So this is technical variation. And so this is a very common situation for many technologies, including mass spectrometric investigations, that we have two layers of variation, is the biological variation and technical variation. And so our objective, so this is important. So this is not really observed, right? It's a conceptual kind of abstraction. Even this is not observed, because we do not know exactly which proteins and how many are in the samples. The only thing we observe is this, is the readout from the mass spectrometer. And so the process of statistical inference is to take what we observe and use it to make conclusions about what we're actually interested in. So statistical inference is taking what we see and make conclusions about what we don't see, but what we want to know. And so if this is our process, then we have two potential dangers here. One is that our conclusions here, or what we see here, are systematically different from the truth. And this is called bias, right? So when what we see here is systematically different from what we want to know. And bias is the worst kind of problem, because bias is reproducible. So if we make some systematic mistake, we will repeat the experiment, we will make the mistake again, and we will say, hey, we reproduced our results. But our results are repeatedly wrong. So this is really the worst problem to have. And then the second danger here is inefficiency. Inefficiency means that our data here are so noisy that for all practical purposes, we cannot say anything meaningful about what we want to know. And so statistical experimental design has as an objective to minimize the bias and minimize inefficiency. And this is really the most important thing, because once the data are collected, if some biases or inefficiencies are built in, I can tell you right away that no amount of statistical sophistication can correct for that, because this is a fundamental flaw with the experimental design. And it's really the most important that all these issues are addressed at the design stage. So let me just kind of show a little bit of notation. I promise I won't do much math, so just to kind of show you again how we think. So just to kind of give you the convention, so usually in statistics we use Greek letters for things that we do not observe, right? So here we say there is a mean protein abundance among all healthy subjects, and this is the mean protein abundance among all the disease subjects. So they're mu because they're not known. Here we have the unknown protein abundances in the individuals which we selected, right? And again, they're in Greek letters because we do not observe them. What we actually observe is written in Latin letters, and these would be different peaks, right? So different fragments of different peptide ions of the proteins, right? Across subjects. And so what we will do, we'll make some kind of summary, and we will try to use this summary to conclude, to make conclusions about what we want to know. And so in this case, bias means that this is systematically different from this, and inefficiency means that the variability associated with our summary is too large. Okay. And so what we're trying to do is to use statistical experimental design to minimize this and minimize this. And this can be done using three fundamental principles of experimental design. And this is something which I did not invent. So this has been around for, I don't know, at least over a century, but probably even more, right? And so what I would like to do now is to just walk you over the strategies which we can use to minimize bias and inefficiency. Okay, so the first principle of experimental design is replication. And I hope I do not need to convince you now that we need to have replicates in our studies, but let me just kind of remind you why we need to do this. So we need replicates for two reasons. So one is to actually distinguish random variation from systematic changes. And the other is to increase the information content in our data and minimize variability. So here's an example. So let's say we're doing a DIA experiment, and let's say we just have one peak per protein, right? So, or one fragment. So that's all we have for the moment, right? And so let's say we have no replicates. So we have one healthy subject and one disease subject, right? So we have one value here and one value here. Let me just kind of say upfront, right, that we are looking at the things on the log scale, right? So I will always talk about the difference between the means, but you know that the difference of logs, it's the same as the log ratio, right? So if you are used to think in terms of ratio, so difference on the log scale is the same as the log of the ratio between healthy and disease. Okay, so we have no replicates. We have this difference between the groups. Is it systematic or is it an artifact of random chance? What do you think? Well, exactly, right? Who knows, right? Nobody knows, right? Because we don't know how much random variation is there. And that's exactly the point. So if you have only two samples, one from healthy, one from disease, you see something and you do not know if it's meaningful or not. So now if you are in this situation when your replicates are very tightly scattered around the mean, then you know that this is not likely to be an artifact. But if you're in this situation when your replicates are very broadly distributed around the mean, then it is very possible an artifact of random variation. Exactly the same difference between the groups, meaning that exactly the same fold change. So I think the first point to make already is that the fold change cutoff makes no sense whatsoever. Right. So this is really important. And the second thing is minimize inefficiencies, because if we increase the number of replicates, then we can be very confident in this mean, very confident in this mean. So even if we have large variability, potentially we can still claim systematic change if we have enough replication. And so because we have two layers of variation, biological and technical variation, we have two types of replicates, right? Biological and technical replicates. And all of this is meaningful. Now, if we have really well-established workflow where we know how to identify the analytes in the spectra, I will give you the upshot is that biological replication is the most important. So if you have a fixed number of runs available, you will be much better off to collect different biological replicates as opposed to measure one person really, really well, but it's still just one person. Okay? So the second principle of experimental design is randomization. And this, you know, I think everybody probably have heard about this. I'm quite sure. However, I am still surprised how often people do not randomize. They think, yeah, I know in principle it needs to be done, but I have all these good reasons why I didn't do it in my experiment. And again, this is really wrong, right? And so let me again continue this particular example. Let's say it's DIA, right? So 1 peak per protein per person. And so let's say that we have all of our subjects here from the healthy group acquired on the first 2 days of the experiment. And here we have all the subjects from the disease group acquired on the second 2 days of the experiment. And so now we see a systematic change between this group and this group, but this change can be attributed to two factors. One to the disease group, but the other to the time of data collection. And there is a systematic change, all right, but is it change we're interested in? Is it really change due to the disease, or is it just change due to the date when we collect the experiment? And the point is that we do not know. And we will not be able to tell this apart. And so in statistical language, it's called confounding. So the two sources of variation are confounded. And maybe you're thinking, oh, maybe I can normalize and so on. So every adjustment for this type of problems requires assumptions. And oftentimes these assumptions are extremely strong to the fact that your conclusions reflect your assumptions a lot more than the data. And so it is much better practice to avoid that. And so here is a example. So when we now randomize our subjects across all the days, so now both disease groups are roughly equally spread across all the days of data collection. And therefore, well, now we have a lot more variability because the variability around the mean has two sources now, right? The biological variation in the samples, but also the day-to-day variation in data collection. But at least we do not have the bias, right? At least we know that this change now cannot be due to the time of data collection because both groups had the same dates, right? So now we know that this change is really due to the status of the disease. And so again, because we have two layers of variation, biological variation and technical variation, we need to randomize both things. We need to randomly select subjects from the population, and we need to randomly allocate our samples to all steps of sample processing, sample handling, and data acquisition. And of course, the comment that I often get is, well, it's not always possible. So if it's not always possible, you need to be extremely critical to other potential sources such as this, which can confound your conclusions. And what it means is that when you summarize your results, you write a paper, or you present your conclusions somewhere, you essentially need to have a disclaimer that all the differences that you find, you think they're due to the status of the disease, but they can be due to other things which you could not prevent. And so essentially, it means that you need your extent of your claim may be much smaller than what you would like. Okay, and so some examples here. So this is a manuscript from a long time ago from statisticians that I really appreciate. So they were at that time at MD Anderson Cancer Research Center, and they do a lot of consulting work for clinicians in their institution. And so they had a manuscript where they essentially collected all failed studies that they had to deal with. And I think it's really nice that they did this. It takes some courage to actually show this type of thing. So it's very old. It's a CELDI example, but it doesn't really matter for our point. So it was a study of 5 types of cancer. And because it was a consulting work, they were blinded, so they didn't know which subject has which type of cancer. And they did all the right things, you know, pick, normalized, you know, everything you would expect from that. And because there was no supervision, they were blinded, so all they could do is discover patterns, right? And so they did this dendrogram, which essentially clusters together subjects in the space of those mass spectrometry peaks. And so what they found was that they had 6 groups and not 5 groups. And they were excited, thinking maybe they found a new subtype of one of the cancer types. But then the next thing they did was they made a plot where the x-axis is the same order of samples as in this dendrogram, but the y-axis is the time of data collection. And yet you see very clearly that these clusters group perfectly with the day of collection, right? And now, of course, the problem is that some of the days they had one type of cancer and the next days they had the next type of cancer. And so this is an example of a study which is completely wasted. So there's no way to save the study at this point. So this is one example, but this is the worst. So this, well, at least they noticed that, and they didn't publish this, they just showed what's going on. But this is a worse situation. So this is in Genomics. The paper is from 2007, Nature Genetics. So researchers studied differential expression between Asians and Caucasians. And they found that 78% of genes are differentially expressed. So 78% of transcripts have different abundance between Asians and Caucasian subjects. That's a lot of genes, right? 78%. And so, which is kind of stunning. So, and this is taken from the blog by Jeff Leake. It's a very prolific blogger talking about different types of statistical issues, especially in high-throughput data analysis. And so he actually and his collaborators, they were really interested in following up on that. So they wanted to take the data and reanalyze this to see if it's really true, and other people wanted to reproduce it. So luckily, so this was done with the Affymetrix microarrays back then. And luckily, the data, the raw data, were made available by the authors. And so they tried to repeat the analysis. They essentially saw the same conclusions, which was surprising. And they couldn't find anything wrong with this data set until they looked at the very raw cell files of the Affymetrix microarrays. And the cell files, they have a timestamp in the corner. So you can only get the timestamp if you get to go to the very, very raw data. And what they found is that there was 1 year difference between when the Asian subjects were profiled and the Caucasian subjects were profiled. No wonder there are changes. You know, all kinds of things can happen in 1 year, right? And so, again, this was a claim which went all the way to Nature Genetics, which essentially has nothing to do with reality because they were looking at the differences in their hybridization efficiency as opposed to differences in the biology, right? So these are real effects, and it can take us in the wrong direction for a very long time if we don't pay attention to that. Okay, and so here's the quote, which is also frequently cited. To call in the statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment died of. right? But you cannot really correct this anymore, right? So I have one message for you. If everything else I say today makes no sense, it's too complicated, talk to a statistician before collecting the data and before designing the experiment, because you do not want to find yourself in this post-mortem situation. Okay. So this is number 2. And I think replication and randomization, this is something which you can do yourself, right? So these, or at least technically speaking, these are not complicated things. So the third principle of experimental design is a little bit more technical, and this is in particular where a statistician can be really helpful in terms of the design. So blocking helps 2 things. It also helps both bias and inefficiency. So let's go back to this example with the day effect where we randomized the samples to different days. And we made a comment that now we have a lot more variation in our data. Some of this is biology, but some of this is day-to-day variation, right? And so if we have a lot of variation, it makes it more difficult to claim that the change is systematic, right, and not an artifact. So it's a problem, right? Plus, if you have a really small number of replicates, even if you randomize, it's still possible by random chance that you have healthy are in earlier days, right, and disease are in later days. So when you have many replicates, things kind of average out, right? But when you have small replicates, you can still randomly, it can still occur like that. So blocking is essentially a restriction on randomization, where you don't randomize everything, but you impose constraints. And so here what we can do, we can impose a constraint where we will say, I want to acquire 1 healthy and 1 disease subject on day 1. And then on day 2, again, I will impose a constraint that I have 1 healthy and 1 disease. On day 3, again, I will have a constraint, 1 healthy and 1 disease. Right? So you still randomly select a healthy and randomly select a disease. You still shuffle which goes first for data acquisition and so on. But within each day, you systematically require representatives from each disease group. And so it has 2 benefits. One is that now you are sure that you have exactly the same number of healthy and disease on each day. So it's good when you have small number of replicates. But also, what now you can do, so you see that there's a lot of variability between the points. But within the same day, if we consider difference between protein abundances within the day, we see that they all point to the same direction. So within the day, disease is always higher than healthy. And because it's the case, it points to differential regulation. So essentially, instead of comparing the mean of this group to the mean of this group, we can calculate the differences and ask ourselves if these differences are systematically going in one direction. So this is easy to draw because I have 1 health and 1 disease. What if I can do 4 samples in 1 day, right, or 10 samples in 1 day? So there are statistical techniques which deal with blocks larger than just 2 subjects, right? So that's not a problem. It's just easy to show on the cartoon. And so blocking requires 2 things, that you design your experiment with blocking in mind, so you have a restriction on randomization, but then also that the data are analyzed according to the blocking design, right? So here it means looking at the differences and comparing differences. And so this is where a statistician can really help you, because you need to know how the data will be analyzed to design the experiment in the first place. And so you will be much better off if you have essentially a working relationship with someone who will be working with you throughout the whole process. I see this way too often when people say, oh, I have some data, can you analyze it for me? You know, we should have talked earlier, right? Because we could have designed the experiment in a way that the analysis becomes easier, but maybe more effective, and so on. And so, you know, this is another kind of misconception where people sometimes, I heard it somebody saying, I don't like to work with statisticians because they ask for replicates. Right? Come on, right? But true, right? But we can do more than just ask for replicates. And so this is one example where with smaller number of replicates, you can allocate your subjects to blocks, and you can perform the analysis with the blocking in a way that increases your efficiency without necessarily increasing the sample size. So given the fixed amount of resources, you can still gain efficiency of your experiment. And this is really important. Okay, and so this is one example. So day is kind of one thing. So from the same paper, what they did here, so another experiment like this, so also blinded. So they had 2 types of cancer, also cell death also pick, normalized, and so on. They found 2 groups, so kind of consistent with 2 types of cancer. Of course, when they unblinded it, what they found is that this is the protocol change, right? And protocol change, you know, so it may be silly, but it may be unavoidable. For example, you know you will need to have a new batch of materials halfway through the experiment, right? Or you need to have a new operator halfway through because, you know, for various reasons or something, right? So this is another example where blocking can help, because what a good way, if you know that you will have to change protocol halfway through, a good way to do it would be to have the same number of healthy and disease here, same number of healthy and disease here, analyze them separately and integrate the comparisons, right? So this would have saved the experiment. And if all of these are healthy and all of these are disease, again, it's in a confounded experiment. Okay, and so another aspect, another aspect of blocking, so blocking has multiple names actually. So blocking is a general technique, but remember we have 2 layers of variation, right? And so we can block by similar characteristics of the subjects, or we can block by technological aspects such as time of data acquisition, right? So now if I focus on the blocking with respect to biological characteristics and with respect to biological variation, this is sometimes called matching. And this is also a term used in clinical trials and in other types of biostatistics investigations. So remember we were saying that we have 2 populations, right? And they have the same characteristics, except that one is healthy and the other is the disease. It's true, but within this population we have subjects which are quite heterogeneous, right, usually. And so age and gender in particular, they affect everything. So usually you really want to make sure that you control for that. And so if your study has a small number of replicates, it can happen by random chance, even if you randomly select from these populations, that you end up in healthy group with all the male and old, and in the disease group all the female and young or something like that, right? And it is effectively confounded, right? And so matching, so blocking, it imposes restriction on how we select subjects from this population. So you can say, okay, I want to have 1 young female representative from each group, okay, randomly selecting that. Now I want to have 1 older female from each group, right? Now I want to have 1 younger male from each group. And maybe you can have, again, 1 older female. So now red and blue groups have the same number of young subjects, the same number of old subjects, same number of males, same number of females. So the difference between the groups cannot be attributed to age and gender anymore, right? And so blocking imposes this balance, right? And again, if age and gender become important, you can analyze it separately for each subgroup and combine the results. And so here's an example of what we did with one of the studies. So this was a collaboration with Munich Heart Center quite a while ago, and it was the first mass spectrometry experience of our collaborators. So what they do, they store plasma samples of everybody who comes to the center for heart problems. And so they had essentially a really large repository of samples, and so they tabulated them like that by different disease groups and also by groups of age and gender. And so this was the original number of samples available for their study. And so, of course, it was a very small pilot experiment. They could not profile all of this. So how can we select subjects from this repository? And so what we would do is, okay, let's take the first group. So there's an older female. So we want to make sure we have the same number of samples from each disease group in this group. And we will randomly select them from what's available. And then we have older male subjects. Again, we will have the same number. We will randomly select them from what's available. Now, you notice that those rows, they don't have to have the same numbers, but every group in a row has to have the same number. So that's the idea. Okay. Any comments or questions? Okay. Nothing to say? All right. Then, well, maybe later you will have some questions. So let me now talk a little bit about the data analysis. And probably most of you have heard of t-test and p-values and things like that. But again, I find it again and again that it's really important to go over this and make sure we all agree on what it does and on the definitions, because it is really important for the next things. So this is just to remind us, right, what we try to do. So we have two groups, right, and we have the biological variation, we have technical variation. We want to summarize our data, right, and we're interested in making conclusions about the unknowns. So let me just more formally tell you what is happening on the log scale, right? So log or no log, and so ratio or difference. So this is also something which we kind of have to talk about a lot. So especially in analytical chemistry and in mass spectrometry, we're used to ratios a lot, right? But the problem with the ratios is that they require two components. And so if we have more than one replicate from one group and one replicate to another group, so now you need to figure how to put together those ratios. What happens if one of the two components is missing, right? And how to combine the ratios and so on. And also, from the statistical perspective, ratios don't have such good properties that you can deal with them. However, sums and differences have much better statistical properties. So it's much easier for us to deal with the numbers on the scale, which allows us to consider differences and sums. So if we want to talk about ratios and we want to talk about fold change, so in this context, what it would mean for us that we will have some typical value in the first group divided by the typical value of the second group, right? So if we kind of go back to this two groups, right? So this is some typical value in one versus typical value in the other. If I take the log of that, so log of the ratio is the difference of the logs, right? So that's exactly what we do. But now, that's not actually what happens, right? Because what we will be doing, we will be taking logs of the individual peak intensities and attempt to summarize that. So what does it mean? So here is the mean of the replicates in the first group minus the mean of the replicates on the second group. So I'm just like spelling it out in a super detailed way. So here is the mean of the first group minus the mean of the second group. So now y is the log intensity, so I'm now say it's the log of the original values. And so logs have the property that sum of the logs is the log of the product. So this can be written like that, and it can be written like that. So essentially what it means, and this is really standard in many areas of statistical analysis, is that if the data are such that we're interested in fold changes and ratios, we can take the log, work with summaries and differences on the log scale, and it would mean that our typical value in group 1 is the geometric mean of the peak intensities on the original scale. And typical value of group 2 is the geometric mean of values on the original scale. So just to kind of to make it clear, right? So everything I will talk about now and later is we take logs of the individual peaks, and then we can take it back and talk about the fold change on the original scale, but it means that the typical value of each group is the geometric mean of the observed intensities. Okay? Just to kind of make it clear. Okay, so this is for the log scale, right? So now that we established that we have a log scale, what we want to do is to find if this difference between the means is more systematic than as expected by random chance. So we need two things. We need to quantify the difference between the means, and we need to quantify what is the associated variability to see if this difference is larger or smaller than random variation, right? And so what we can do, and again, our question is we want to know if the two means in the population, right? So we want to know this, but we observe this. So we don't know what the means in the population are, but hey, we can make guesses about them. So let's just make a wild guess that between the two groups, there is no difference in means. We'll just state that, right? And so this is what is called the null hypothesis. And then if this wild guess is wrong, then the alternative is that there is a difference between the groups. Okay. So now we want to have our signal. So we'll essentially be building a signal-to-noise ratio. Our signal is here the difference between the two groups, right? So this is the difference between the groups, where this is the mean of the first group, this is the mean of the second group. So the only thing which is left now is to understand what's the associated noise with this ratio, right? And the noise associated with that is the variability in the first group and the variability in the second group, and then we also divide by the sample size. Right? So this S1 here is the sample variance in the first group, S2 will be sample variance in the second group, and we take the square root. Okay, so now the question why. So let me just spend a little minute on that. So this is sample variance in the group, but this quantity, sample variance divided by the sample size, is the variability of the mean. And taking the square root of this is the standard error of the mean. Okay, so these are really important terms. So now let me go to the next slide. I will show you what I mean, and then we will come back here, okay? So every once in a while, I take figures from this series of vignettes. Nature Methods publishes these vignettes, like 1 or 2 page articles called Points of Significance, which talk about very focused small things in statistics. And so I'm taking figures from that, but there's also text that comes with that, so you can actually look at these vignettes. Okay, so what we're trying to do is to characterize the variability associated with the means in each group. So how does it work? So let's say we have one group of subjects, and their protein abundances come on the log scale from this type of bell-shaped distribution. So now what we can do, let's say we take 3 subjects out of this distribution randomly, calculate their mean. Okay. Then we will continue, we'll take 3 other subjects from this population, calculate the mean. And we will take 3 other subjects and calculate the mean. And then we draw the histogram of those means. So this is what this histogram of the means is. So it essentially tells us how often the means from 3 subjects here versus there are similar to each other or different. And because the subjects have natural variability of protein abundance, it means that their averages will also have some variability, right? And so this distribution or this histogram of the means is the sampling distribution of the mean, right? And so the broader this histogram, the more uncertainty we have in the mean, because from one replicate to another, the more mean of our subjects will vary. But so now, if instead of 3 replicates we take 5, we see that this histogram becomes narrow. 10, it's even more narrow, and 20, it's even more narrow. So as the number of replicates we take from the original distribution increases, our histogram of the mean, so the sampling distribution of the mean, becomes more and more narrow, meaning that we become more and more confident in the mean. The means become more and more similar to each other, right, no matter which replicates we take. And so the variability of this histogram is the variance of the sampling distribution, and the square root of this is the standard error. So the standard error is the standard deviation of this histogram. So it tells us how confident we are in the mean. So this is the standard deviation of the original population, but the standard error is the variability associated with the summary of the data, right? So with the mean. And so now when we build a signal-to-noise ratio, so what we're doing, we're saying, okay, this is the difference between the means. This is how much variability we have on the first mean. This is how much variability we have on the second mean. We combine them together because we have 2 means, 2 groups, and then take the square root to have it on the same scale as the original mean. So this is our signal-to-noise ratio associated with the difference between the 2 groups. And so an interesting fact is that we can start with something else. We don't have to start with the bell-shaped distribution. We can start with some other distributions and repeat the same exercise. And what happens is that when the sample size is large enough, the means always follow this bell-shaped distribution. So if you start with something really weird, so you see that this distribution is wider than this distribution, right? So there's more uncertainty. So it takes more replicates for it to become very narrow and bell-shaped. But eventually, even if your data are not normal, the means themselves are approximately normal. And this is called central limit theorem, that the means will become roughly bell-shaped. And so a question that I often ask is, okay, well, what if my data are not normal? Well, if they're normal exactly, then this is normal. If they're not normal, then they are approximately normal. So if we have enough replicates, so we can assume that the means are roughly normally distributed. So this is another use of replicates, that it also protects us from deviations from this kind of nice bell-shaped patterns. So now we have all the pieces, right? So we have 2 groups, so we build the signal-to-noise ratio. And the very last piece here is that we need to know essentially how large is large, right? So how large the signal-to-noise ratio have to be to claim that the difference between the groups is larger than the associated variation. And so this is kind of the beauty of working on the log scale and the beauty of working kind of in this framework, because if our wild guess is correct, if we know, if in fact there is no difference between the groups, if the data are normally distributed, and if we build this construct that we can analytically, without seeing any data, we can say what is the plausible range of the signal-to-noise ratios that we can expect. So, so here, so this is what we just did, right? And so if in reality there is no difference between the groups, if the data are normal, and if we build the signal-to-noise ratio, then we can predict exactly the distribution of the signal-to-noise ratio that we will have. And so if the data are not normal, then it will be approximately bell-shaped based on the central limit theorem. And so this is why it's very useful, because now what we can do, we can say, so you know these distributions, the area under the distribution is the probability that the event occurs. So now if we take alpha, let's say 5%, and allocate half of this in the lower tail and half of this in the upper tail, then essentially what we can do is to say, well, let's take the signal-to-noise ratio that we have. If it falls in the middle of this distribution, we say, yeah, well, that's exactly what we expected, right? So there's no contradiction with what we assumed, that there is no difference between the means. However, if we calculate our signal-to-noise ratio and it falls very far in this tail or very far in this tail, we will say, that's really different from the expected distribution of values which we thought we will see. What's wrong? Well, what's wrong is that we assume that there is no difference between the groups. So this is evidence against the null hypothesis. And so the cutoff is based on this alpha value, which is the false positive rate. And so we know that we will have, let's say, 5% of probability of rejecting the null hypothesis when in fact the null hypothesis is true. So this is our probability of false positives. Does it make sense? Yeah? And so if it is normal, then it's exact. If this is not normal, then it is approximately normal, right? And so this is why it's much easier to work on the log scale, because it is very difficult to have any analogy of this expected distribution of some constructs if we work with ratios. And so now we can say, okay, this is how large we would expect these values to be, and if they are too small or too large, then we reject the null hypothesis. And so the last piece here is the p-value. So the p-value is just another way to do the same thing. So I can take my observed signal to noise, I can overlay this on top of this expected distribution, and I will overlay it here, and I will because it's symmetric, I will also overlay this in the mirror image on in the other side. And so now the area to the left of the minus observed value and to the right of the plus observed value is the p-value. And so if the p-value is less than alpha, it means that it is outside of our rejection region, so we reject the null hypothesis. And if the signal to noise is here, it means that the p-value is larger than alpha, and there is no evidence against the null hypothesis. So that's all that we really do here. Okay? Does it make sense? Okay? So the very last concept associated with that, and this is also important, especially when we start doing things like sample size calculation and so on, is the concept of statistical power. So as I just mentioned, this is, again, I adapted this from this vignette. So this is just another picture of the same thing, right? So this is our distribution. So this is called the reference distribution, which we expect to have when the null hypothesis is true. So let's say here we just consider one tail. Let's say alpha is 0.05 in one tail. So we'll expect upregulation in group 1, right? So we don't really care about the second group. And so this is the formula for signal to noise. Now, if in reality there is a difference between the groups, so this value will be much larger than 1, right? Because this group will be much larger than this group. So it will be shifted. So you can actually draw this. So this is what happens when the null hypothesis is true, but when in fact the alternative is true. So this numerator is larger than 0, right? And so the whole thing shifts to the right. And so if the null hypothesis was true, this tail will be the probability to have a false positive, but that's our decision when in fact the alternative is true. And so this blue area to the right of the decision rule is the probability of rejecting the null hypothesis when in fact the alternative is true. And so this is the probability of detecting the difference when it exists. And so this is statistical power. And so this is a false positive when in fact the null hypothesis is true, but it is the true positive, so power, when in fact the alternative is true. And so we see that the signal-to-noise ratio moves to the right if the alternative is true. Now, what are our strategies to increase statistical power, to increase the probability that we detect the true difference? Well, one thing is easy, right? So we can make false positives larger, right? So if we allow more false positives, right, so then, yeah, we will have more true positives, right? So this is the trade-off. So if we increase alpha, so our rejection cutoff is more liberal, so of course we increase the power as well, right? But this is probably not a good way to go, because the price of detecting more differences is also that we have more false positives. But another strategy to increase the power is to take this distribution and move it further to the right. So what will make it move further to the right? Well, of course, if there was the true difference between the groups larger, it will be more to the right, right? But also if the sample size is larger, right? So it's in the denominator of denominators. So making sample size larger makes this whole quantity larger, meaning that it also shifts the whole distribution to the right, and it increases the statistical power. And so this is how, as a preview, how we will go about sample size calculation. So how far to the right do we need this distribution to be, given that we fix type 1 error alpha? Does it make sense? Okay, so this is for the hypothesis testing. Now, the problem with hypothesis testing is that, or with p-values, is that it essentially gives you yes or no decision, right? So differentially abundant or not differentially abundant. So it doesn't really give you a characterization of how big is the amount of change, and it doesn't really give you, you know, kind of direct characterization of uncertainty associated with that. And so another way of summarizing the data, which is equivalent to hypothesis testing but gives a different type of information, is to work with confidence intervals. And so let me just tell you a little bit about, so essentially error bars, right? And so so let me first define the confidence interval for you. So confidence interval, it's actually not an interval itself, it's a procedure. And it's a procedure such that if you keep collecting the data and applying this procedure, which gives you a lower and upper limit, and you keep collecting the data over and over and calculating lower and upper limit, then 95% confident interval will be such that 95% of times you apply this procedure, the interval will contain the true mean, and 5% of times the interval will not contain the true mean. So confidence interval is not the interval itself, it's a procedure, and the confidence is the property of the procedure. So out of 100 repetitions of this procedure, 95 will contain the true value, but 5 will not contain the true value. So it has this probabilistic aspect, and so the confidence interval, the whole procedure, is actually derived using exactly the same signal-to-noise ratios that we discussed before, right? So this is our signal, the difference between the two groups. This is our noise, which we used to have in denominator when we did hypothesis testing. And these critical values, they are exactly our bounds of the decision rule. And so these bounds are such that the area to the left and area to the right are exactly alpha, right? And so we just rearrange essentially this equation, and because these quantities have the probability alpha in both tails, then we can show that this type of interval will have exactly this property, that 95 out of 100 will contain the true value, if all the assumptions are verified, right? And so, again, that our data approximately normal, right? And also that our subjects are independent from one and randomly selected from the population. And so now, if we wanted to draw error bars, so it's kind of useful, right? Because now it kind of tells us both the magnitude of change and the uncertainty around this. And so now we have 3 ways to draw error bars. So if we just want to, if we have one group, so we can do 3 things. We can have the group mean plus minus standard deviation. we can have group mean plus minus standard error, and we can have a confidence interval. And so what I often see is that people will just say, I have an error bar, but they will not qualify what type of error bar they have in mind. And the visual appearance of these error bars and the information content of these error bars is actually very different depending on the meaning of the error bar. And so here's an illustration. So for different sample size, you have two colors. So the light gray is the standard error of the mean, just to remind you, right? So this is the standard error of the mean, right? So how much confidence we have in the individual mean. And the dark color is the confidence interval obtained by this procedure. And you see that the confidence interval is much wider than the error bar, which shows standard error of the mean. Well, both intervals become narrower and narrower when the sample size increases because we have less and less uncertainty in the mean, right? But the appearance of these error bars is dramatically different depending on if it displays confidence intervals or standard errors. And so just to kind of highlight that, so this is again from this series of vignettes. So what they did here, they took 10 replicates and they adjusted the variability such that you have exactly the same appearance of the error bars for two groups. This error bar shows standard deviation, this shows standard error, and this shows confidence interval. And so the p-values of comparisons are very different depending on the meaning of the error bar, which looks the same. And here they did the opposite. They fixed the p-values and they showed different types of error bars. And so you see that standard deviations can be quite large. We can have quite a bit of variability in each group. And it's okay because we are comparing the means. We're not comparing the populations, right? Now, standard error of the mean has to be fairly small and fairly separated, right? Because the individual means have to be fairly stable, right? We need to have enough confidence in them. But the confidence interval can actually overlap to some extent to give us the p-value of 0.05. So the point here is, at the very least, when you do show error bars in figures, at the very least qualify what the error bars you have in mind. And the second thing is that the expectation of the results actually will be quite different depending on the type of the error bar. And here they also show how for different p-values, so what the standard error bars look and what the confidence interval error bars look like. So, and you see that for the standard error of the mean, they really have to be quite different, right? But the confidence intervals can tolerate quite a bit of overlap in general. Okay, so this is the hypothesis testing and p-value. Nothing to say? Very silent. It's very clear. Oh, okay. All right. So in this case, we have 15 minutes now that I can use to tell you. So I told you how good p-values are, right? So now let me tell you how bad p-values are. It's all about the balance, right? So we need to make sure that we know all the problem associated with the p-values. So, okay. So p-values are used a lot, obviously, right? But they're also somewhat abused sometimes. I don't know if you have heard, last year, a journal in psychology banned the use of p-values in their research because they felt like people were abusing the p-value reporting and the p-values were derived incorrectly and so on. And so this generated a lot of controversy, and especially now with the high-dimensional experiments and so on, to the point that February last year, the American Statistical Association issued a statement on statistical significance and appropriate use of p-values. American Statistical Association have never issued any statements about anything. So it was the first time when they felt compelled that it's really important to provide, the guidelines of what p-values can be used for and how and what they should not be used for. So it was a really big landmark for the society. So they had 6 points. So the first point is p-values can indicate how incompatible the data are with the statistical model. good enough, right? So we had our model, which states that there is no difference between the groups. So if our observed signal to noise is in the tail of this distribution, it means that it's incompatible with our initial guess, right? So we reject the null hypothesis. So this is good, right? So that's exactly what we talked about. P-values do not measure the probability that the hypothesis is true. This is a very common mistake. You have a small p-value, therefore we have low probability of null hypothesis. It's completely unrelated, not in this type of framework. So if you know about Bayesian statistics, so in Bayesian statistics, the hypothesis itself is random and it has a probability. What we discussed here, hypothesis has no probability. It's a fact of life. It's either this or that. It cannot be maybe this, maybe that. It's either the protein is either regulated or not regulated. It cannot be regulated sometimes and so on. So it's not the way that this thinking is going. So we cannot interpret the p-value as the probability of the null hypothesis. But that's the next thing. Scientific conclusions and business policy decisions should not be based only on whether a p-value passes a threshold. So you could not use small p-values. You cannot make small p-values drive everything in your life, right? So that's essentially what it is saying, because there are many reasons for that. So one can be, remember we were talking about essentially comparing the means, and if we have a lot of replicates, we can be very, very confident in each mean, right? And so essentially we can drive statistical significance by increasing the sample size infinitely. So essentially, and I will show you in a minute, we can pretty much make anything significant if we have a large enough sample size. Is it a good decision? Based on a statistical conclusion, not necessarily, right? And so the next point is proper inference requires full reporting and transparency. So, for example, if we have confounding, right? So if we have two groups, remember we had healthy on the first 2 days, disease on the next 2 days, we can perform testing just as we discussed, and we will have small p-value. But this will not be a correct conclusion because there is a confounding, and the reason for the difference between the groups is other than what we're interested in, right? So without full reporting, we will not be able to understand that. And the example was the gene expression study, right? So if you do not report fully that Caucasians were acquired 1 year after Asians, so you would not be able to understand that small p-value was essentially an artifact, right, of the experimental design. So this point is very important for us, I think. So the p-value or statistical significance does not measure the size of an effect or the importance of the result, right? So you can have any fold change. It can be statistically significant. Is it practically important for us? Maybe, maybe not, right? So maybe, for example, if it's a screening experiment and you have a long list of proteins with small p-values, maybe some of the small fold changes, maybe you're not interested in them. Maybe some proteins, they don't come from the right pathway, right? Or maybe some proteins, they don't come from the, I don't know, cellular versus extracellular and so on. So maybe you do not want to follow up on everything, right? So practical significance is not the same as statistical significance, and you really need to use both. So essentially, use common sense in addition to statistical analysis, right? Which is kind of, important. And the last thing is a p-value does not provide a good measure of evidence regarding a model or a hypothesis, right? So if you have a small p-value, it doesn't mean that all of our assumptions actually hold, right? We need to question our assumptions separately. So this is one kind of thing, just how to use this, right? But the next thing is what I want to also emphasize, that p-values are derived from the data, right? So we build the signal to noise, right? We overlay this on the distribution and so on. Our data are random, right? We just talked about the biological variation and the technical variation. P-values are a function of the data. Therefore, p-values are also random. So if next time you repeat your experiment and you collect new data, well, guess what? You will not have the same p-value. And this is also very important to remember. And so this is an example which comes from this Nature Methods publications, not the vignettes, but it's also the same series. So they took two populations. It's like a simulated example, so just computer generated. They have two populations, and the difference between the groups is very small, 0.5, compared to the variability in each population. So what they did was they took 10 replicates from group A, right? and 10 replicates from group B. They did exactly what we just talked about. They calculated the p-value. P-value is 0.005. The difference is 146. But then they repeated it again. They took 10 other points from population A and 10 other points from population B. Look at this, p-value is 0.8. So here, you will probably claim it's statistically significant. Here, it's not still significant. And look at the fold changes. Here, it's much bigger than the truth. Here is in the opposite direction of the truth. And this is because our data have variation. So the conclusions are also random. A third time, fourth time, you see that it can vary quite a bit. And so this is also important, right? So your small p-value today does not mean small p-value tomorrow. And so we have to be careful about that. But so now they take it to the extreme. So they continue the same example. Instead of repeating it 4 times, they repeated it a very large number of times, maybe 1,000. And they just plot the histogram of p-value. They plot it on the log scale so that we can emphasize the region of small p-values. And so now sample size 10. They take 10 replicates from each group. And so you see that a very small, 186 out of 1,000 instances had small p-values. The majority had large p-values. So small sample size of 10 gives you very small percentage of small p-values. Sample size 30, so now you have 469 instances with small p-values out of 1,000. Sample size 64, okay, now 800 instances with small p-values out of 1,000. And it only takes sample size of 100 to have 948 instances with small p-values. So sample size essentially does affect the stability of our results. And so, well, that's why statisticians ask for replicates, because our results are much more reproducible and much more stable if we have more replicates. And so if you have, and this is very typical, right? For example, in DIA experiments where we have 3 or 4 or 10 replicates, well, we should be fully expecting to be in this situation, where many proteins, we may or may not be detecting things, right? So it depends on the difference between the groups and the amount of variation. And so they have a separate representation of this. So here the x-axis also log p-value, the same thing as here. But the interesting thing is the y-axis. So this is the this is the estimated fold change. And so you see that, so this is 10 replicates, 30, 64, and 100. As the sample size increases, so the p-values start moving towards smaller values, right? And the fold change kind of moves towards 0.5. But what I find really fascinating is this histogram. So what they do here is they plot the estimates of fold changes, but only among comparisons which survive p-value cutoff. So they take 10 replicates from each group, calculate the p-values. P-value is less than 0.05. It contributes to plotting the histogram. Otherwise, it doesn't contribute, right? So this is only the histogram of fold change among statistically significant instances. And look at that. The true difference is 0.5. Every single one of them is above 0.5. So when the sample size is small, not only our conclusions are unstable, but when we do claim significance, the extent of regulation is dramatically exaggerated. So the fold change is actually much larger than what it really is. And so now as the sample size increases, so little by little this histogram moves down, and so only with 100 replicates, it fluctuates roughly around 0.5. So this is another reason why replication is important. And so they do the same thing with confidence intervals. Confidence intervals have the same property, right? Because it's derived with the same way. Okay, and so just 2 more things which are also important. So all of that I talked about for the whole hour and a half essentially focuses on working with one protein, right? So we take a protein, we calculate signal to noise, we derive the p-value, we understand that the p-value has limitations and so on. The fact is that we don't have one p-value, right? And especially now with DIA experiments, we have thousands of proteins, right? Each of them has to go through the same procedure. And so now we also need to account for the fact that we repeat this over and over, one protein after another, and this also contributes to artifacts, right, and some variability of our conclusions. And so the motivation for this also from Jeff Leake was the fMRI study. This was a poster where somebody used fMRI to look for a brain activity of dead fish. And they found many active brain regions in dead fish. Why? Because they tested different locations one at a time, and well, if you test enough, you always find something. Remember we had alpha, right? The probability of false positives, which was at 5%. So if we test 100 regions, we have 5 false positives. If we test 1,000 regions, we have 50 false positives, right? If we test 10,000 regions, we have 500 false positives. So we will always find something which, in this case, is obviously wrong, right? And so this is the same thing, of course, for DIE experiments. And so let me just illustrate this in a different way, the problem of multiple testing. Let's say we only have 2 proteins to make it simple, right? And so for each protein, we do exactly the same thing, the t-test, right? So this is protein 1. So this was our reference distribution, right? We allocated tail alpha over 2 in each tail. So now if the null hypothesis is true, if there's no difference between the groups, so like in dead fish, right, nothing is changing. So this would be the region where we will make false positives, right? And we will do this regardless of whatever happens with protein 2, right? So now this area plus this area is 5%. We can do the same thing for protein 2. Now we have the reference distribution. This is our rejection region. This area plus this area is 5%. But our study has both proteins, so what really happens is this, right? And so now the combined area together, well, it's more than 5%. And so now you can extrapolate this to more and more proteins, so essentially the probability of finding at least 1 false positive is essentially 1. So we'll always find something, right? And so this is the problem. So when we have many proteins, what we have discussed so far is not enough, because we also need to account for the fact that we do many of these tests. And so one way to go about that is essentially change our decision rule and push our decision boundary further away, so that now the area outside of the dashed line is what we want it to be, let's say 5%. And it's fine. So now what happens is that for the individual protein, we become more conservative. So we have to have much stronger evidence for differential abundance to control the overall error rate, right? So what does it mean to have stronger evidence? So either it's only proteins which are more regulated, so we lose proteins which are marginally regulated, or we need to have larger sample size, right? Remember power, right? We need to have more power for that. So that's the problem, right? So essentially, if we have more proteins, we will be more conservative, or we will need to increase the sample size. And so if you have heard of Bonferroni approaches, that's exactly what they do. They take alpha and they divide this by the number of comparisons, right? And so it pushes our decision boundary further in the tail. But if we work with DIA experiments and we have 10,000 proteins, we take our 5%, divide by 10,000, and it's extremely conservative, right? And it's very clear, because the best way to avoid false positive is not to find anything, right? So then for sure we don't make any false positives, right? So it's not quite why we're doing the experiment, right? And so a change in statistical literature occurred kind of probably 20 years ago by now, where we essentially said, let us change the criteria. When we have multiple hypothesis testing like that, let us change the criteria and not control the probability of making at least 1 false positive. Let's say, okay, we're fine to have some false positives, provided that we control the proportion of false positives in the whole list of differentially abundant proteins. And this is the false discovery rate. So what I talked about so far, it was the family-wise error rate, when we want to have very small probability of making at least 1 false positive. Here we say we're fine having some false positives, but let's control their proportion, let's say to 5% or 10%. And so this is a classical table. So this is the true number of proteins which are not differentially abundant. This is the true differentially abundant proteins. This is our decision. We claim not differentially abundant, we claim differential abundance. And the letters here, these are the counts of proteins in each situation. So as before, these counts, they're not observed. The only thing that we observe is how many total rejections we have. And so Bonferroni or family-wise error rate, they want to claim that the probability that V is anything except 0 has to be very small. But false discovery rate says, on average, the ratio of V to R has to be as specified, for example, 5%. And because V can be non-zero, and we want to control the ratio of V over R, it allows us to be less conservative. And so just like with the t-test, I was saying that we can analytically show what is the expected range of our signal-to-noise ratios. There are procedures which can show essentially how to control false discovery rate, even though we do not observe those for numbers. And so examples would be using the approach by Benjamin and Hochberg, or there are also two-group mixture approaches by John Story or by Brad Efron. So I don't have time for that, but essentially there are ways to control false discovery rate. And so the output for DIA, and this is what we do in our tools, would look like that. So this is a very typical output. It's called volcano plot. So the x-axis will be the log fold change between the two groups. And the y-axis is p-value adjusted for false discovery rate. And then we take minus log of that. So that now, because we're interested in small p-values, so essentially the top left corner and top right corner mean statistical significance, and the x-axis, you can think of this as practical significance, right? So how big fold change you want to entertain. And so you can also have the vertical cutoffs, this is practical significance, but the horizontal cutoff, for example, of 5% means that your false discovery rate outside of the cutoff is 5%. So we will come back to this on Friday. So I just see that we need to stop now. So let me just take 2 more minutes to do one more thing, which I think is extremely important. So what I talked about so far is really fairly technical things, right? So how we kind of design the experiment, right? Randomize, use blocking and so on, and then how we build the signal-to-noise ratios, calculate p-values, multiple testing, right? And so that's all important, provided that we have a very clear protocol of how we go about all the steps of experimental design, data collection, and analysis. But there are so many decisions that go through this, which can potentially make us deviate from the protocol, that all the technical things, all the mathematical things I talked about essentially may be violated for reasons other than math, right? For reasons of the decisions we make along the way. So here is also a nice comment from Nature from a couple years ago. So essentially, here's what happens, right? This is our overall workflow. So experimental design, collecting the data, cleaning the data, modeling, summary, p-value, and then we don't find what we want. Our favorite protein is not in the list, and we blame p-value, right? But maybe something else is happening, right? Maybe some other things which affect reproducibility other than just, all the steps that we talked about. And so let me just give you some examples here. So a very big danger for this type of analysis, which has nothing to do with mathematics, is deviations from pre-specified protocols. So here's an example of a clinical trial. So they were developing study for an antidepressant. They considered the trial was designed for some types of outcomes. Turns out none of the outcomes became interesting, so there's no change. However, they also tried a whole bunch of other outcomes, and one of the outcomes showed a significant effect. So said, oh, look, the drug works for this. So we tried all of these things that we designed it for, didn't work, but we tried many more things. Now it works for something. Let's market the drug for this, right? So we just talked about multiple testing, right? So something is bound to happen just by random chance. Because they changed the protocol and they decided that we did not test for this, we test for this. They decided to market the drug. Eventually, they realized it was an artifact of multiple testing, right? So you cannot change the way you cannot change your purpose of the study based on the data from the study, right? Here's another one. So allocation of subjects to groups. So this was a study where they wanted to look for differences for women at peak and off-peak fertility cycle. So arguably it's not an obvious thing to do. So what they did, they essentially kept changing which woman is in the peak cycle versus off cycle. And so they moved women between groups long enough to conclude that at peak fertility, women are more likely to wear red or pink shirts. So same thing for us, okay, we have two groups, right? Oh, you know what, this sample doesn't look good, let me exclude it, right? Or now this sample actually it was mislabeled, let me put it somewhere else. And we can move our samples enough to essentially get to any type of conclusion that we want. You can still go through all the motions of doing t-test and p-values and so on. It's not part of the mathematical argument. But these things are incredibly dangerous because we can essentially arrange our data to the point that the p-values would look like we want them to look, but doesn't mean make them correct, right, or reproducible. And so just to finish, so this is what sometimes in psychological science called researcher degree of freedom. So it is not the data degree of freedom, but it's the researcher degree of freedom. So how much we change the protocol so that the data look like what we want them to look. And so here is the manuscript entitled Undisclosed Flexibility in Data Collection and Analysis Allows Presenting Anything as Significant. Right? So it is not mathematics, but again, it's a little bit of a common sense. And so the right way to do this is to, before collecting the data and before designing the experiment, is to develop a overall protocol which you will follow no matter what. And you can say things such as, I will use this procedure for quality control. I will remove samples if I see this and this, right? I will use this procedure for peak detection, peak quantification, and so on. I will, I don't know, use filters like this and like this, right? This approach for statistical analysis. And then no matter what happens, you stick to that. So this would minimize the artifacts due to, overfitting and overusing the data, but it would contribute to reproducible research. So takeaways. So what we talked about is defining the problem, right? Translating the biological or clinical goal into statistical goal. Experimental design. Avoid bias and inefficiency using randomization, replication, blocking. Follow a predefined analysis protocol. Not alter the design, not cherry pick parameters, and understand state limitations if the full kind of randomization and so on was not possible. And document all the steps in terms of the executable code. Okay.
Great.
Sorry for going over time.
Thanks a lot, Olga. Are there any questions now for Olga? Or she's also here on Friday, so can ask that. Maybe I can ask one. So I was curious about this simulated data set. You had these two distributions, and you had these 2D histograms with the fold change and the p-value, and the thing was moving over. So is the reason that the fold change was shifting only because you had a p-value filter? So if you look at the fold change estimates raw, these would be still centered on
correct. So it's really the bias of the p-value cutoff. And so for the p-value to be small, you really need to have exaggerated fold change, right? Because the sample size doesn't really help you, right? So essentially, it's all coming down to the statistical power here, right? So what you want is to move this distribution to the right. So either you increase sample size, but if your sample size is fixed, so when the apparent fold change is large, so then you'll be able to see it. So yeah, so this is really the bias on the p-value.
Okay.